Dr. Gina Agistratetu of the Helmsley Charitable Trust and Dr. Kurt Griffin of the Sanford School of Medicine at the University of South Dakota will discuss a program to screen for type 1 diabetes and intervene in the progression of disease. Medical science may be on the brink of an unprecedented accomplishment, detecting which babies are at risk to develop type 1 diabetes before it becomes clinically evident. This could allow for early intervention aimed at delaying the onset and even prevent the clinical manifestations associated with this autoimmune disease. Research that has enabled this remarkable achievement has been steadily advancing over the years. Now it is time to put scientific theory to the test. But the kind of clinical trial required takes dollars and especially time, while millions around the world continue to live with a life-threatening disease. That is where public, private, and academic partnerships can accelerate our ability to improve human health through innovation. A new program designed and funded by Helmsley Charitable Trust and Sanford Research is precisely the kind of collaboration that could make type 1 diabetes prevention a reality. Here's how it's working.
I'm Kurt Griffin. I'm a pediatric endocrinologist at Sanford Health, and I have the privilege of directing the clinical trials for the Sanford Project, which encompasses all of our type 1 diabetes research. One of our new and exciting programs is our PLEDGE study. This is general population screening for type 1 diabetes risk and celiac disease. This is a disease that we have a treatment for, which is insulin, and 100 years after the discovery of insulin, we still don't have any way to take care of the underlying problem that leads up to type 1 diabetes, which is autoimmunity. Type 1 diabetes is an autoimmune disease that develops silently and over a prolonged period of time, and we are now getting some evidence that there may be some ways where we can slow it down if we know who's on that path to getting it. Unfortunately, many of the children who present with new onset type 1 diabetes do so at a very advanced stage when they are in a state called diabetic ketoacidosis. Even that single episode of ketoacidosis at diagnosis is associated with worse blood sugar control for some years afterwards, and there's evidence that it actually has effects on brain development just from that single episode. Most of the families that find themselves in that situation had no idea that this was coming on. We now have ways to be able to predict and detect early who may be heading in that direction by measuring autoantibodies that are relevant to type 1 diabetes. These are antibodies that are markers that the immune system is starting to attack the pancreas in a way that it really shouldn't be doing for the rest of us who are healthy. So we're now at the point where we're beginning to get a little bit of traction against the autoimmunity. And one of the things that we've been thinking for a long time, and now we have our first evidence, is if we can treat this earlier, we may be able to stop it, or at least slow it down before it gets to the point that a child is actually sick and needs insulin. In order to do that, we need to know which children are on that path and who might actually benefit from that intervention. Diabetes TrialNet has done a wonderful job screening family members of people that already have type 1, but the math of this works out such that the vast majority, 90% of the people who are going to get type 1 diabetes don't already have in the family and they're not eligible for these family screening programs. So in order to be able to catch these kids before they get sick, we really need to expand the screening so that eventually we can offer this to every child. So the PLEDGE study, like many, has taken a while to get to this point. It is something that really, I think, leverages what is special and different about Sanford as far as being an integrated health system. And from the time that I arrived at Sanford in 2013, I've already been talking about how might we be able to move to this eventually. And then there's the serendipity of, who one meets at meetings. And in 2017, I happened to be in the takeaway line for lunch in San Francisco behind Jenny Thomas from Australia, and we got to talking about her ideas for screening and what might be able to predict who goes on to develop the autoimmunity. And we started developing a project from there, and that's where the PLEDGE name originally took hold. And if we fast forward 18 months, at the same immunology of diabetes meeting, this time in London, I sat down with Ann Korolova from the Helmsley Charitable Trust and began to have a deeper discussion about the overlap in mission and interest and ability to do this project at Sanford, matching with the interests of Helmsley in funding these sorts of programs.
I am Gina Giustratidou. I am the director of the Type 1 Diabetes Program, and our goal is that we want every person with Type 1 diabetes to thrive in life by supporting grants for better access to care and devices, and also we support research and technology for a better tomorrow. We had discussions with the team at Sanford and Dr. Kurt Griffin for a couple of years. As you know, we have a common interest on Type 1 diabetes. And then I would say 2 years ago, we had the pleasure to go and visit the Sanford team and Dr. Griffin in South Dakota, and where we are tried to identify what we can do together to fight this disease. It's better to be proactive than reactive, and Pledge offers this possibility, offers to screen your children, and offers, if the children have the increased risk for Type 1 diabetes, they can provide meaningful care and meaningful education for something that can really change their life. I strongly believe that what the outcomes of the Pledge project, they will be very beneficial for the Type 1 diabetes community.
There's clear evidence that the children who develop Type 1 diabetes at younger ages, many of them already have antibodies certainly by 1 or 2 years of age. To measure autoantibodies, we need just a tiny little bit of blood. We're trying to match our collection schedule with when children are tending to come in for vaccines and sometimes are routinely having blood drawn already. In that case, we're able to piggyback on the blood collections already done and just get a little bit extra for the Pledge study. While the Pledge study is designed to capture children starting at birth, we are welcoming enrollment of any child up until their 6th birthday.
My name is Allison Gates, and I live in Sioux Falls with my husband and 2 kids, and this is Lily, and she's participating in the Pledge study. I was really interested in having Lily participate in this study because my sister had been diagnosed with Type 1 diabetes when she was really young, and I saw some of her struggles and how it kind of affected her life, and if there was any way that I could learn more about it or help my kids if they were predisposed to Type 1 diabetes, I really wanted to do that. It's just a simple finger prick to kind of collect the blood, and that's what they would be testing, and then we would find those results. So it seems like, a quick errand that can really give us a lot of information that would be important for the future. And so it seems definitely worth the very minimal effort that we had to put in to participate in this. So the parts of Pledge that are really innovative have less to do with the assays that we're using and more with how we are implementing them, and the extent to which we can integrate them into the routine care that children are already coming in and getting. We really try to leverage an established workflow, so we're not trying to invent something new, but rather take advantage of what's already happening every day. The autoantibodies are analyzed in batches, so once we get enough samples, they're shipped all together to our collaborator in Seattle who runs the autoantibody assays. These are being performed on a CLIA-certified platform, so the results are clinically actionable. Fortunately, the vast majority of these are going to be negative. For those few people that do have a positive result, we will reach out to them by telephone and ensure that they understand what the positive result may or may not mean, and then connect them and be sure that we have a way to monitor them going forward so that if somebody is on that path to diabetes, we have a way of catching them before they get sick. If and when they get to the point where they need insulin, we can start that early before they get very sick and before they come in ketoacidosis.
That by itself has tremendous impacts on how well they do after the diagnosis, as well as obviously saving the costs of an intensive care admission. Right there is a significant impact financially as well.
We're trying to assess if oral insulin, for example, can prevent type 1 diabetes. Our vision is that this platform, it will go beyond type 1 diabetes. We will be able to study other autoimmune diseases, other childhood diseases. We want to use this little drop of blood to study 10, 50, 100 diseases. I'm asking you to unite with us because prevention is a very, very difficult and complicated effort. I'm asking you to unite because we all want a life without disease.
Type 1 diabetes isn't a short-term diagnosis, it's a lifelong thing. Being able to educate yourself as a family, I think, is very valuable. There's not a lot of things you have to do. Like a finger prick, that seems pretty doable, right? We can do that.
The data that come out of Pledge are exactly the sort that are going to be necessary to assess whether this is ready to be moved into routine standards of care for all children. The Helmsley Charitable Trust and other organizations are already talking with the groups that establish standards of care for children in order to learn what is necessary to be able to bring population screening for type 1 diabetes into those standards. So the opportunity to be contributing to something that could touch every child in the United States and beyond is really awe-inspiring.
A personalized approach to rare diseases is on the rise. John Crowley, chairman of the board and CEO of Amicus Therapeutics, sets the scene and hosts a conversation with Dr. Michael Yeaman, Dr. Joe Weimer, and Dr. Catherine High.
Hi, everybody. Good day. I'm John Crowley, and it's my pleasure to host this session on a personalized approach to rare diseases. I am very excited about where we are today in the field of genetic medicine. I go back to the late 1990s when two of my children were diagnosed with a rare form of muscular dystrophy known as Pompe disease. And we were told then that there was little research, there was nothing that could be done, go home and enjoy the time that we had with our children. To us, that wasn't an acceptable answer, and it launched us into the world of medicine, into the field specifically of rare diseases and genetic medicine. And thankfully, with the intervention of a treatment, our children's lives were saved. But it also launched us into this field, realizing that with more than 7,000 rare diseases, there is so much unmet need. And we have to marry that against the promise of some tremendous science. In the last couple of decades, we've seen that science in small molecule precision medicine, in biologics, now increasingly in gene therapy, and someday soon the promise of delivering medicines with gene editing. So we're very excited about what the future holds, and particularly excited to be able to host this today with the Cura Foundation and with the Pontifical Council for Culture. I was reminded of some of the words of the Holy Father, of Pope Francis, back in 2019 on the World Day of the Sick, where Pope Francis described what he called a selfless love for every human being without distinction of language, culture, ethnicity, or religion. That's what we see in our world of genetic medicine. These diseases know no geographic boundaries. And a couple of themes that I'd like to see come out in our great panel ahead. One is the notion of access. We are developing great medicines increasingly. We also need to make sure that every person in need, everywhere in the world, has access to these new great medicines. That means more approvals. It means a commitment for companies and entrepreneurs like ourselves to make sure that our medicines are fairly priced and broadly accessible. And in those parts of the world where they can't afford these medicines, to make sure that we get them to patients in need. It also means that we need to continue to think about the regulatory pathways to realize that this is a battle against time as much as it is against the mistakes of nature. So ensuring access, but also another point I'd like to see to come out in this panel is continually to make sure that patients are at the center of everything we do and everything we think about. In developing medicines, it really is this virtuous circle. It begins with early stage research, oftentimes in research centers and universities. It moves on to entrepreneurs and small companies. It's partnered with venture capitalists, investment bankers, people to bring the capital necessary to do all this great research. Our hospital systems, our physicians, our nurses, regulators, people ensuring access again along this virtuous circle. And when we get to the top of the circle and we realize that we've made a medicine that's extended and enhanced life, very rarely is it a cure. And we need to do it again until that day where we truly have cures. But again, at the center of all of that really is the notion of the patient. And how do we consistently have that mindset that when we make decisions, we need to think, if you had this disease, whatever it may be, where you were the mom or a dad of a child, what decision would you make? That's, I think, the heart of being patient-centered, that mindset. And it's something I'm very excited about because at the end of the day, ultimately what we are delivering is hope to patients in need. And it has to be real hope, tempered with reality. But with the great science that we have today, I'm increasingly excited about being able to deliver on that hope in the decade again. So if I began with a quote for Pope Francis, I'll end with a quote that I think frames this conversation ahead well. Pope Francis said that only if we see ourselves not as a world apart, but in a fraternal relationship with others, can we develop a social practice of solidarity aimed at the common good. So with that, my hope is that we can advance the common good in this great discussion. So I'm pleased to kick this off and welcome our panelists. I am joined today by Dr. Michael Yeaman of the UCLA School of Medicine and the Guthrie-Jackson Charitable Foundation. Also joined by my colleague, Dr. Jill Weimer, both of the Sanford Research Institute, as well as Senior Vice President at Amicus Therapeutics, and also by Dr. Kathy High, President of Ask Bio. So we're going to have this conversation. We're going to talk about rare diseases and talk about some of the great advancements that we're starting to see, building on years of research and experience, I know, for each of you. So Michael, let me begin with you. When you think about the study of rare diseases, paving the way for personalized and customized healthcare where no one size fits all, how do we think about it? How do you think about it?
John, thank you very much. It's great to be with everyone today. You know, our work with the Guthrie-Jackson Charitable Foundation focuses on a rare disease called neuromyelitis optica, or NMO. And NMO occurs when the immune system misrecognizes normal proteins in the central nervous system as though they were foreign, reacts to them, leading to inflammation and loss of myelin, which in turn can lead to vision loss and loss of mobility and other issues. And what's so interesting about NMO is that even though the patients share a similar immune disorder, the disease can manifest in many different ways and can have vast differences in each patient journey. So in this respect, we appreciate each person, in each case, in and of themselves, and we always approach the solution as treating the person, not the disease. And so what we're really trying to do is find the best intersections between personalized medicine and precision medicine. And, you know, over the last decade of work, we've accelerated the science of NMO and advanced the clinical medicine and diagnostic accuracy. And we've gained credibility with industry and brought a bunch of expertise and perspectives together in a way that after 100 years of no approved therapy for NMO, in the last 2 years, we now have 3 approved therapies that again find the intersection between precision medicine and personalized medicine. So John, I think rare diseases really can open doors to many other solutions.
Now, Michael, that's a great point. I think, people ask me from time to time, why do you work in the rare diseases? There's so few patients versus much more prevalent disorders. And part of what I share is that by unlocking the biologic secrets of understanding human genetic disease, we will unlock the secrets to much more prevalent challenges of human health. So in one hand, there are many rare diseases. Taken together, they're as common as all cancers combined, or more so. But then also when you start to unlock some of these biologic secrets, particularly in the neurologic field, you start to understand better diseases like Parkinson's, Alzheimer's. In some cases, a lot of the diseases of neurodegeneration really are just a series of human genetic diseases. I know in the CNS field, increasingly, children who, maybe over the years were diagnosed in broad categories of epilepsy or cerebral palsy or even autism, increasingly are being genotyped and realized to have single gene or sometimes multiple gene mutations. So it is an exciting field in that regard. Jill, let me turn to you. A lot of opportunities to help people. Where do you think the greatest opportunity lies in using genetic targeting for rare diseases?
Absolutely. Thanks, John. I think, just building off of what Michael mentioned, over the last decade, last 5 years, last 2 years even, we've seen just a blossoming in the genetic medicine field. And I think a lot of this is built off of standing on the shoulders of giants such as Kathy and leaders in the gene therapy development field, and starting to recognize that it's not one size fits all approaches. I think many times I've heard it's simply as simple as copy and paste to restore a gene into a patient suffering from a rare disease. But we know that that's not the case, that each person has unique genetics, and even if they have mutations in a similar disease or gene, it can result in very different diseases. One of the examples I think that we're seeing are in diseases like frontal lobe dementia, where you have mutations in progranulin that can also lead to a pediatric disease, one of the forms of Batten disease. And so similar approaches can be used, but just looking at the unique genetics and what are the things that you need to be able to supply to that patient. So looking at personalized approaches, some of the pioneering work that's being done in antisense oligos to be able to customize those therapies to a unique patient's genetics, I think are really helping us turn the corner to developing personalized medicines, but also just not thinking about just the wild type gene. Are there things that we can do to enhance gene therapy approaches or genetic approaches to get better cellular targeting, to get better distribution within a cell, get it to the right places and get it turned on and functioning more efficiently? So I really think the next couple of years are going to be just remarkable to watch in how these technologies unfold in genetic medicine.
Great, thank you, Jill. And you're right, Kathy, you've been one of the great pioneers in this field. You have been. When you look today, most gene therapies fall into two categories where we've seen approvals, and there have been very few. Single gene disorders that you're familiar with, and then also in the CAR T cells for leukemia or lymphoma. Where do we see the next approvals?
Well, John, I think there continue to be exciting programs for other single gene disorders. I would think of the hemophilias. I would think of Duchenne muscular dystrophy and other muscular dystrophies as well. So I think that we will certainly see more approvals for single gene disorders. And, if you look at Zolgensma, for example, or Luxturna, these are AAV gene therapies that were approved on relatively small data sets, 40 or so patients for both of those. I think that what that tells you is that it testifies to the dramatic effect in a single gene disorder of putting the gene back into the right physiologically relevant target tissue. The clinical results are dramatic, they're clear-cut, and you don't need a very big trial. Most of the effort, in fact, comes around CMC, not around trying to figure out what you're seeing clinically. And I think that those kinds of dramatic results have fueled an interest in trying to develop gene-based strategies for complex acquired disorders. You mentioned a couple of those, Alzheimer's disease, Parkinson's disease, and so forth. And I believe that you're right, that what we're learning from the study of single gene disorders gives us lessons that are important to carry over into complex acquired disorders. And I do believe that there will be successful gene-based strategies for a number of those disorders. But I think in the near term, what we will continue to see are approvals for single gene disorders. But I'm excited about the future for a wide range of diseases.
Yeah, I think we all are. And when you think about, you got in the United States, you and Jeff and the team at Spark oversaw the approval of the first gene therapy in the United States, then followed by Zolgensma for spinal muscular atrophy in 2019, and there haven't been any since. And you brought up an important point on CMC. So that's manufacturing. That's the analytical tools to make sure that we understand the quality and the quantity of gene therapy vectors that we're delivering. And it's a really important point too, that yes, that basic biology and science is very hard, but we make great progress. And when they work just profoundly. But we've got to get the manufacturing right. We've got to get these analytical tools right. In some respects, perhaps the entire field got a little ahead of ourselves. And I think together as an industry, we're working through with regulators, making sure that we've got the right quality of materials and have the tools to understand the quantity. In gene therapy, I think the process, the manufacturing process is the product. And we've got to get that right. So terrific points. Michael, maybe if I turn back to you, is there an opportunity to use platform technologies to treat many of these diseases, or do they all need to be, bespoke or custom tailored and developed one at a time? How do you see that?
Yeah, John, I think there are opportunities. There will be some new and remarkable opportunities to apply, let's say, the power of the immune system, for example, to solve many seemingly different diseases. For example, in NMO, we're using tolerization to reset the immune system in a way that takes an autoreactive disease and essentially tames the immune system so it no longer reacts to proteins that are in us that would otherwise be normal. And the point here is that we can use the power of the immune system to deal with any protein antigen that might be the cause of an autoimmune disease. So in that sense, it's sort of theme and variation. And this could apply to any autoimmune disease for which an autoantigen is known. And the other side of that same coin might also be true for other diseases that are related to immune dysfunction. And I'll just use the example of cancer, which we often think of as, cells gone rogue. And that part is true, but cancer emerges because the immune system fails to detect and destroy these cells. So even if the earliest possible detection, cancer cells have already figured out how to evade and outpace the immune system. And I think what we can do is use sort of inverse tolerization to prepare the immune system for cancer cells it has yet to see. And in that sense, prevent cancers that might have emerged otherwise.
Yeah, very exciting, Michael. Jill, let me ask you. There are a lot of promising advances in technologies for rare diseases, particularly in the gene therapies. What are you seeing in your work at Sanford or Amicus or the field more broadly?
Absolutely. I think one of the things for me personally that I've experienced over the last 5 or 6 years is as scientists, we're so often trained that we have to go it alone, that we have to come up and conceptualize the ideas and put those into practice. But I think if we can actually work in partnership and as teams, and teams for me has been working with family patient foundations, really helping them drive the direction that we're going and get input on drug development, partnership with industry. Really, I think we can move mountains. I think for so long we've tried to do it alone as individuals, and I think we need to look for unique ways that we can actually partner. And I'm not saying this just from the context of paying lip service to collaboration where maybe pharma sponsors a particular project in an academic lab. I think it's really getting the thought leaders together into a room, bouncing ideas off of each other. One of the things that I look back to in the neuroscience field from a decade ago was the Brain Initiative. And the Brain Initiative was not intended to fund neuroscientists like myself. It was actually intended to fund computer engineers, structural biologists, physicists to come into the neuroscience field, use their tools that were very unique from an individual neuroscientist trained to synergize and really move the brain mapping process forward. And I think in drug development for rare diseases, we can learn a lot from that process. and really accelerate what we're doing by bringing in expertise from folks that maybe we wouldn't normally collaborate or partner with.
Great, thank you, Jill. Maybe Kathy, back to you. You highlighted a few moments ago one of the challenges that we're addressing as a field, and that's manufacturing. One of the other challenges is safety broadly. We need to make sure for these medicines, particularly in gene therapy, that they are both effective and safe. What do you think about the recent safety events in the field and how they limit development? Maybe your thoughts on technologies to address it.
Well, John, before I tackle that, I just want to echo Jill's comment. Drug development, and particularly gene therapy drug development, is the ultimate team sport. And it really puts a premium on communication of innovative ideas and just playing as part of a group. Nobody can do that sort of work alone. So I do want to put safety into context. I think that many people know that over the last year, we've learned as a field about a number of safety events across the board in gene therapy. Some in lentiviral mediated gene therapy in hematopoietic stem cells, some in AAV trials where the vector was infused either intravenously or given neurosurgically to the brain. And it's important to consider all these things in context. If you look at the number of new investigational new drug applications submitted to the FDA over the last 5 years in the field of gene therapy, in 2017, the number shot up pretty dramatically to over 100. The next year, it was over 200 new investigational new drug applications. And the following year, it was even more. So there's been a dramatic increase in the activity in the space. And so I think it's important to recognize that in the context of greatly increased activity in the space, many more trials, many more people enrolling in trials, that yes, it's not surprising that in that type of setting, we begin to learn the limits of what can be done with these vectors safely. But I also think that we're looking at some issues related to, for example, who can enroll in these studies. And can a person with liver disease expect to safely be injected with a vector into the liver? Or, so I think we're learning some limits about what we can do. I think that part of what we're seeing is simply a consequence of many more trials going on. The good part about that is that the pace of learning accelerates. And the challenging part is that we will be learning some limits.
I think that's very important. And also, too, to remember as we look at this, there is no cookie cutter approach. I think we have to look at each molecule and each disease and each patient population. Again, bring in that patient perspective. What risks are they willing to tolerate for a chance for a much improved and longer life? So that risk-benefit assessment lens is going to be important for all of us, whether it's developers, we as innovators, regulators, to think about and to continue to get that patient perspective. Michael, when you think about gene therapy broadly as a field, yes, we have come a long way to go. How do you see that fitting into the therapeutic arsenal, and in particular, how it may differ from other kinds of interventions that we're more traditionally used to?
Right, John. I think as Jill and Kathy have emphasized, we need to sort of blend the promise of sort of genetic modifications with the need for safety and targeted strategies. And so, for example, in immune-based disorders, including autoimmune disease, infection, cancer, transplant rejection, and others, there's usually more than one immune system component that is a little bit out of balance, either working too hard or not working hard enough. And gene therapy can be used to deliver what we call gain in function solutions, where we turn up the immune system a bit, for example, in cancer, or it can be used to deliver loss in function solutions, where we turn things down. And examples might be, for instance, in NMO, there in many patients is an extra set of copies of genes involved in the complement system. And you can imagine targeting some of those extra copies by either gene editing or deletion strategies that can potentially turn down the autoreactive status of the immune system. The same is very likely true for the genes involved in the molecular handshake between cells like T cells that govern the immune response and other cells that present antigens to them in a way that can diminish autoreactive responses.
Great, thank you, Michael. Jill, back to you. When this wonderful science, but ultimately we have to translate it into and through clinical studies. Right now, clinical studies are still very complex. How do we think about clinical trials for rare diseases, ideas for accelerating evaluation, reducing costs? What are your thoughts?
Yeah, I don't think I need to tell this panel that one of the caveats that we face in the rare disease space, particularly in pediatric rare diseases, is that if we follow the normal time course for a traditional clinical trial, many of the patients will have deteriorated and succumbed to the disease in the time that we get our ducks in a row and process through a phase 1, phase 2, and phase 3. And I think traditionally the time to complete that safety to efficacy to the phase 3 trial, there's the gaps in between, the processes to analyze that data to get approvals to move to the next steps. So one of the things that I think you're seeing more and more discussed in the rare disease space is this process of seamless clinical trials, where you would just evolve within your clinical trial to phase 1 to phase 2 to phase 3 without those gaps. Adaptive clinical trial design, where we're learning as we go along, right? We're racing as fast as we can to get these medicines to the patients as needed. And we're learning as we go along. So the ability to adapt our clinical trial design after it's initiated, built upon learnings that we experience as a part of that clinical trial. And that could be as simple as changing the primary outcomes, secondary outcomes, changing how we're analyzing the data as we're going through the process, I think is also going to lend to the rare disease space. And then as Michael had touched on, sort of these platform technologies and looking to our comrades in the cancer space to look at trial designs like basket trials, where we might be looking at multiple different drug indications within one disease, or umbrella trials where we're looking at multiple different disease or genetic indications with the same platform, I think really is going to be the only way, John, to speak to how we accelerate these processes and how we cut costs to be able to move these drugs towards the market and towards patients.
No, those are great points, Jill. And I really think about it as this truly holistic system. If we can reduce the barriers to bringing more medicines to patients, we'll bring the cost down, we'll increase competition, we'll increase innovation. So again, in that notion of a virtuous circle, I think that's incredibly important. I would just add, as we think about the use of biomarkers in this studies, I think those will be increasingly important. Use of natural history studies. We all get calls from patients and family members on a daily basis asking about what they can do in their diseases, oftentimes very, very rare diseases. And what I usually tell people is try to help us find more people living with your child's or your disease so that we can help them when we do have therapeutics, but also so we can begin tracking the natural course of their disease to build natural history studies. Because in many of these diseases, we just cannot, it's just not acceptable to use placebo controls. You can't do it in diseases like Batten disease that we work in, or other rare, devastating disorders. So we have to be creative. And that also means thinking about biostatistics. Traditional biostatistics are likely not to apply in many of these therapeutic areas. So to think more creatively about using the adaptive studies you described, Jill, but also using tools like Bayesian statistics and multi-domain responder indexes, looking at the entire totality of the data in advance. And that's going to require all of us, again, to think about this in that benefit risk assessment. And again, against the notion of time. How do we beat time as much as we beat nature? Maybe, it's a big question, Kathy, but I'll throw it to you to conclude. What do we think the future of gene therapy holds?
Well, John, I think that if we look at the history of other new classes of therapeutics And I think about monoclonal antibodies. They were not products when I was in medical school. And I remember that there was a lot of controversy around issues about using monoclonal antibodies as therapies. The first one was approved in 1986, OKT3. We still use it in transplant. The next one was not approved until 1994, so a long gap. And then 3 years later, Rituxan was approved along with another monoclonal antibody. And the year after that, 4 more were approved. So it took more than a decade. But by the end of that first decade, the number of products was really ramping up. And by the end of one more decade, monoclonal antibodies were 8 of the top 20 biotech drugs. I think that we're in a similar inflection point now for gene therapy. I think that with all of this activity in the space that I alluded to earlier, will all of those be products? No, probably not. But right now, gene therapies have a better percent likelihood of success than most other classes of therapeutics. So meaning that people are really moving toward things where they have very strong proof of concept in animals. But I think that we will continue to see the pace of licensing, the pace of approvals of gene therapies increase. And so that's one thing that I think. And I think the other thing, gene therapy is really different from other classes of therapeutics. A person undergoes a fairly intensive period of medical management when the gene therapy is administered and in the initial weeks following that. But then the trade-off is that they attain a new and better status of health and well-being that allows them to move forward with far less medical intervention than they might have been accustomed to. And to me, that's very exciting.
It is exciting, again, continuing to move patients along that virtuous circle until there is a cure. And, I hope that gene therapies, my hope is that, the days of a doctor telling parents that your child has a rare disease and I'm sorry, there's nothing that can be done, that's just not acceptable anymore. And I hope that the future brings a discussion where a doctor can say, you've got a beautiful child, they have a rare disease, we have a plan, we have a medicine, we have a gene therapy that could provide great benefits and hopefully a cure. So that's the hope that we need to deliver on. So thank you all. This was terrific for your thoughts and most especially for just all the great work you do. You guys deliver real hope. Thank you.
Therapies for rare diseases are being developed faster than ever before. Here to talk about the trend is Frank Sasinowski, Director of Hyman, Phelps, and McNamara, in conversation with Dr. Peter Marks, the U.S. Food and Drug Administration's Director of the Center for Biologics Evaluation.
Hi, I'm Peter Marks, and I'm here today with Frank Sasinowski, and we're going to talk about some issues related to rare diseases. I'm going to ask him a couple questions, and he'll ask me a couple questions. So let me get started here. Frank, you've helped so many companies achieve drug approval for rare diseases. What do you think are the greatest hurdles and what can be done to accelerate the discovery process?
Oh, that's a great question. Thank you, Peter. Everyone who volunteers to take part in a rare disease trial is really a hero by participating. And so it's incumbent upon us to do all we can to get every bit of information from that trial participant. So let me give you an illustrations of a couple different ways we can do that. One, use every control. We're used to looking at just the concurrent control, but it's also important to look at every other way in which we can look at that trial participant's results. If, for instance, we have measurements for a year before they started the trial, we can use patient as their own control, use that. We can, of course, do a match to a natural history control. We can use the patient as their own baseline control. So we have different ways. We can give more weight to the more important ones, the ones that a trialist would regard as more rigorous. But of course, we should include every type of analysis because it'll help inform us as to whether or not the trial participant is truly benefiting. A second thing to do is to combine endpoints. We incense at the altar of the pre-specified primary endpoint when we actually measure an awful lot about what's going on with the patient with all their secondaries. So we have ways through Bayesian and frequentist methods to combine, to take the unique information out of each one of those endpoints so that there's not redundancy, take the unique information, combine them in a way so that we're valuing every bit of information from that precious trial participant. A third thing to do is that each person we know in rare diseases is unique. That is, the way a rare disease expresses itself phenotypically is very heterogeneous. So each person is going to be different. So if what we can do is look at, for each person, look at everything that's measured, all their subjective, objective endpoints, the biomarkers, and regard that in some sort of way and rank it versus the other trial participants, or do a global, like a Likert assessment. So that's another way to recognize the uniqueness of each person. And finally, Dr. Woodcock and I were keynote speakers at a rare disease conference 10 years ago. She said, look, we're a visual society, use videos more. I think that there are ways to do things like do a video at baseline with activities of daily living of that person, and then do it at the end of the trial. Send it off to a central independent expert reader who's blinded to what treatment arm they're randomized to, and let them analyze that data. I think there's lots of things that we can do, and it's very exciting, Peter. Great question. Thank you.
And given that we've been in this COVID-19 pandemic, are there lessons we've learned from COVID-19 that may aid in developing therapies for rare diseases?
Yeah, and I think you've taught us a lot, and thank you for your leadership. And I want to give a shout out to President Biden and Dr. Kessler in the White House. Here's a person who should get a presidential award. But you've taught us through COVID, Peter, that two things, I think, for the rare disease community. One, you've taught us that you should use every piece of information. That's what you did with COVID. You used everything, animal pharmacology data. You used every scrap of information, cohort analyses. So I think we should do the same thing with rare diseases, because I think those people are just as desperate. And I think the other thing you taught us is the way you analyze the data was to look to see whether or not the known and potential benefits outweigh the known and potential risks. Now, I wouldn't want to say potential benefits when we're talking about rare disease, but maybe we should look to see whether the known benefits outweigh the known and potential risks. Thank you.
Great. And kind of finally here, what are we making enough progress on rare diseases, and what are you optimistic about?
Progress. NIH published a paper in the fall that said it estimated at the rate at which we're approving new therapies, it'll be 2,000 years before we have a single therapy for everyone. Most people don't live 2,000 years, so we need to do a lot more. I'm optimistic because our first drug law in the United States was in 1906. In 2016, President Obama, December, the last bill he signed was the 21st Century Cures Act, which recognized that FDA should put patient experience data in every drug it approves. We finally recognized patients. The word patient didn't appear for over 100 years in our US drug laws. I'm optimistic because we're finally moving beyond the paternalistic system we've had in the past. And we're now inviting patients to have a seat at the table. I'm very optimistic. And let me turn the tables on you now, Peter, and ask you some questions. There's several thousand, 7,000 rare diseases. Their small patient base makes it difficult to sign up enough subjects for clinical trials. Do you envision a future in which we have a global regulatory paradigm that can set up worldwide trials for rare diseases and offer a better chance of enrolling enough patients?
Yeah, I think we're going to have to work towards that. We started to work towards that with WHO before this pandemic started. And I think we really have to continue work in that direction. There's no way that we will do justice to people with rare diseases globally unless we come to some harmonization so that advances in any given country for a given disease can be rapidly exported to every other country where those same rare disease patients are located.
Thank you. I'd like to ask the same question you asked of me. Are there any lessons learned from COVID-19 pandemic that may aid in developing therapies for rare diseases?
Yeah, I think I'm going to echo something you said, but I'll start with one thing, which is that one of the things that's really helped during COVID-19 is people coming into the agency very early on and sketching out what they're going to be doing so that they don't have wasted steps. I think that's a really important thing that we can do and take forward. And I would just hallelujah to you on amen on on really using every bit of data we get and not discarding data because it comes in so many different forms, and provided we have evidence that we have benefit on how someone feels, functions, or survive, we should be able to move forward here.
Thank you. That's great. Last Friday, you spoke at the FDA Rare Disease Day, and you spoke at that time of N of 1 therapies. You know, as science evolves, we are beginning to be able to diagnose truly unique inherited disorders that may affect only a handful of people around the globe, maybe even just one. And yet with gene therapy, we may be able to tailor a treatment to just that individual's unique mutation. This raises two issues. One, how do we handle that case? And two, it illustrates what's happening across the entire spectrum of diseases, and that as we learn to identify genetic causes, we are targeting advanced therapies for smaller and smaller populations. There's no end in sight to this progress of science. And what does that mean?
So that's a great question. Thank you. I think this is a place where we have some regulatory framework, but the regulatory framework is not a perfect fit for gene therapies. Gene therapies live in this world where they are biologic drugs, but they have elements of almost a little bit of a device-like to them in that we can reuse pieces of them. An example of that is how we reuse the vector that carries in the insert in a gene therapy. And as we start to have to encounter more and more gene therapies, unless we can find a way that's more analogous to a razor blade analogy where you don't have to reinvent the razor every time, you just change the blade. And that's probably the kind of thing we need to do so that we don't have to reinvent the whole manufacturing process information needed. The idea here would be to hopefully move to a place where we have enough confidence in our razor handle that we understand how to make that. And we understand that as long as you put certain razor blades on that handle, you're always going to come out at the end okay, right? And so that's, I think, the place we want to get here so that we can move through and accelerate it from taking another 2 millennia to get to all these other rare diseases, to be able to move through them in a facile manner. And that's what we owe patients, I think.
Well, let me ask you one more question, and that is that how do we get people across the entire socioeconomic spectrum, including those with lower incomes, participating in clinical trials about rare diseases? Maybe I learned from COVID-19.
I think what we've learned from COVID-19 is if you talk to people in language they can understand about the importance of participating in clinical trials, it doesn't matter from where they are on the socioeconomic scale, they will potentially see the benefit. One has to explain that in language that's understandable, though.
No, thank you. And I also think that COVID-19 taught us that maybe we can move out of academic research institutes into community health centers. We can do that through telemedicine, through remote assistance. We'll be able to reach people. You know, they can't take 2 or 3 buses, or in the rural poor can't have access. So I thank you. I think that's perfect.
The world has many extraordinary men and women who accomplish more than an average human seems to be able to in a lifetime of 24-hour days. The important question for these overachievers is not how they do it, but rather what they do with their remarkable talents. Is it in search of personal aggrandizement and riches, or is it, as in the often quoted aphorism, to leave the world better than he found it? Frank Sasinowski is an individual who's devoted his life to the latter.
We all focus on what's right in front of us because that's right, that's what's most immediate. But when you see yourself as part of a global program, a global movement, you understand how important you are to that system.
Few people have done as much to ease the suffering of millions around the world by working to find and approve treatments for rare diseases. But Frank's path to what he calls his ministry has been somewhat unconventional. An early love of science led to a degree in genetics from Cornell, followed by 2 master's degrees at Berkeley in physiology, nutrition, and public health. That expertise led him to testify to California legislators to improve funding for school meal programs. That's how Frank rekindled his second academic interest, the law.
And they reduced the burdens in the law for accelerated approval. They made the post-approval requirements less onersome.
He realized he was now behaving more as a lawyer. That's when he felt that the Lord was showing him that he could make the greatest difference through the law.
In 2002, it was Frank's scientific lawyering. Yes, there is such a thing as scientific lawyering. Frank has trademarked it.
After his law studies at Georgetown, Frank served a stint in the USDA and the American Public Health Association, again helping improve nutrition. Then he heard another calling, using the law to do still more for society. Frank joined FDA in 1983 as regulatory counsel in the Center for Drugs and Biologics, where he was key to implementing both the 1983 Orphan Drug Act and the 1984 Hatch-Waxman Act, the enabling legislation that spurred pharmaceutical industry research into what had been unprofitable rare diseases. Those laws transformed the orphan drug and rare disease world, changing it from isolated, often fruitless pursuits of treatments to a burgeoning field that has so far produced 500 therapies for rare diseases, with giants in the drug world actively pursuing more. Frank is the recipient of the Lifetime Achievement Award from the National Organization for Rare Disorders, the organization he's helped to become the voice for the millions with one of the estimated 7,000 rare diseases. What drives someone to such relentless, single-minded pursuits? Frank says he has 3 lives: family, professional, and spiritual. But his professional life is his ministry. It's how he sees the face of God in each patient who calls out for help, suffering with a rare disease. For doing God's work, and for constantly striving to heal the world one person at a time, we award Frank Sasinowski the Pontifical Hero Award for Inspiration for his unwavering advocacy for those who live with rare diseases. To talk about accelerating the path to regenerative medicine, Dr. David Pierce, President of Innovation and Research of Sanford Health, joins moderator Dr. Peter Marks, Director of the Center for Biologics Evaluation at the U.S. Food and Drug Administration.
Hi, I'm Peter Marks, and I'm here with David Pierce, and we're going to talk about some of the challenges and opportunities for developing cellular therapy products. I think probably I'll pose the first question to David about, from your perspective, what do you see are the real challenges, the places of need for developing these therapies?
Yeah, thank you, Peter. So I'm David Pierce, the President for Research at Sanford Health, and it's certainly an interesting time with the terminology cellular therapies that's linked to regenerative medicine. So, as the overseer of research for a large health system, the challenge for us is that we have patients seeking these types of therapies all of the time. And obviously, as many of us know, there's many offerings that are defined as cellular therapies, and some may or may not actually be cellular therapies. But, most, if not any of them, are not approved. So as a health system, it's our responsibility to actually work through to legitimize these. And I say legitimize these in terms of the actual science and the application and the outcomes, as well as making sure we adhere to the ethos of first do no harm, because we still have a long way to go. I think during the course of this discussion, I can populate with some information that we believe we're moving in the right direction. But the first challenge of really primarily is people want this, and people are seeking it, and unfortunately getting it without true validation.
Yeah, no, it's an interesting place that's exactly, we're really well aligned there. The whole regulatory framework here for cellular therapies was premised on this concept that certain things would be safe and their efficacy could be assumed. That's why, skin transplants don't need a biologic license when they're not manipulated. They're, particularly, and there are many products that don't need to come in for licensure under our regulatory framework, but ones where we don't know, where we can't predict their efficacy, or ones where there's significant manufacturing, which could introduce safety concerns, that's where we have to get involved. And I think that's been a real challenge for us, because much as people want these therapies, the data supporting the use of many of them is very thin. It's been a challenge because I think it's been a challenge, and I have a lot of empathy for those who are trying to develop them, because unlike pharmacologic therapies, where you have a big pharma house trying to develop these, which has the kind of funding behind it that can, many of the people trying to develop these therapies are individual practitioners or groups of practitioners where they don't have the resources. And so some of the issues can be challenging. And we've been trying to work on those regulatory issues to help provide pathways for people to collaborate and to be able to get the kind of information by any one of a number of means that will help us feel confidence that we have the effectiveness data that says that these therapies actually do what they say they'll do.
Yeah, I couldn't agree more. So, and we are in those trenches. That sort of, I'm paraphrasing you there in terms of, how do we get there? Our patients want these cellular therapies. The first thing was really education, because there's a stigma of stem cells is always, surrounding, cellular therapies. And, in the Midwest where we're located, that really brought to light a few questions initially with respect to when we launched what we called our Cellular Therapies or Regenerative Medicine program. And that was an interesting discussion because, when it comes down to, in an oncology, we've been using bone marrow transplantation for many years. So once you get a comfort level, really, even with some of our physicians, that stem cells or a cellular product has been in use for many years. And then, of course, the next step was for us is to really evaluate the landscape. And I'm going to assume it's the same for many others. I mean, we can talk about cellular therapies in oncology with the sophistication of CAR T or in type 1 diabetes. But our patients have really been seeking, musculoskeletal or orthopedic treatments. Everybody wants, everybody's got those aches and pains. And it seems to me that we have billboards and newspaper ads, internet ads, constantly referring to these types of therapies that could, treat pain in the shoulder or pain in the back or osteoarthritis, which may be generating that pain. So we took a very measured approach to start with, which was just look at the landscape of what was out there. And, much like with bone marrow, we know what the cell lineage is there. A lot of people are offering these fat-derived products in terms of lipoaspirates or whatever. So we looked at the cellular lineage there, took a hard look at the science and really opted to pursue clinical trials in orthopedics initially, using what I would call stromal vascular fraction. And this is an interesting time because, Peter, I really empathize with you because the science just can't keep up with what's actually happening out here right now. Stromal vascular fraction, there's some adipose-derived stem cells in there. There's leukocytes, there's a few other things. Who knows what growth factors and other bits and bobs are in there. And that's really, where other people have been using that, and as you're aware, we've actually performed and completed clinical trials using this fraction now. First of all, just to get the efficacy, so safety, obviously, which I talked about first, do no harm, and of course it's safe ultimately because it's autologous. It's from the individual. You take it out here and basically filter, collect the cells and apply it to the joint. I think the next thing is obviously now we have preliminary efficacy in some of our studies, but not to the level in terms of multi-site clinical trials. And that's where I'm looking forward to with respect to how we advance that. But of course, the general public, first of all, is very impatient and they don't really understand what we need to do with respect to some of these things.
Yeah, no, I think that's exactly right, is that the general public, it's hard to understand. It seems, especially because it seems like they're my own cells, they can't harm you. Why not just do this? the issue is that, there are, if you do enough procedures with autologous cells, you occasionally will come into a contamination or other issue. And any harm, if there's zero efficacy, is a problem, right? So it would be really nice to be able to know that these programs are working, if nothing else, because financial toxicity of therapies that don't work are a problem for people. They're also a problem, even if these are were to be paid for by insurance, they would be a problem for the healthcare system. But right now, for many people who are paying out of pocket, it would be nice to know that they were actually getting the benefit that was more than just some placebo effect of knowing that they were trying something. So I think just to be commended about doing the studies and doing them carefully and correctly and under IND the way you're doing them, that's what needs to happen here. So it's a great example of what needs to happen, and going out and doing what needs to happen takes the courage to do, because the courage and fortitude to do, and you're to be commended for it, because so many just take the easy route of saying, oh, well, we can just inject this, and they're just doing it. And it's not leading to the kind of knowledge that we need to advance people's health and know that this actually really works, and to move from advancing. I suspect that if we really make progress here, we'll see, just like you talked about, it may turn out that if you find some stromal vascular fraction that actually works, the next thing will be to find, well, how do you tweak that? What element of that works? And unless you have the initial data to know that it actually works in the first place, it's going to be very hard to get there. I mean, one of the things that sometimes I think it's important for people to just understand is that, the difference, people say, well, what's the difference? It's a drug. The difference is most small molecule drugs have a couple of atoms that are hung together next to each other, they're basically low molecular weight products, and are not complex macromolecular assemblies like a protein. But wait a second, cells are super complex assemblies, and in some ways they're almost like autonomous beings. They can do things on their own, and so the complexity that we're working with here is so much greater. So in a way, it's kind of an interesting thing that unfortunately the entities that have to work with this are working with a substrate here, the cells, that are challenging to control. And so trying to do what you're doing, of trying to control as many variables as you can and then get an answer, is so important.
Yeah, thank you. I mean, it's interesting. I mean, I'm a cellular guy myself, and the biology is complex. The other thing, which has been the interesting challenge, is, of course, the operations of doing this in orthopedics. Taking a cell. Well, obviously, first of all, doing some imaging. So you need radiology to see where you want to deliver the cells based on the osteoarthritis or the injury, if it was a rotator cuff tear. And then, of course, you're going to be dealing with, your orthopedic or sports medicine physicians who are dealing with the actual patient with respect to the malady. And then, of course, getting that lipoaspiration as well, which is typically done by plastics or, the vascular guys. So we've worked to credential, our sports medicine guys, which brings the cost down considerably as well, but also so we can do more of a point of care. And I think that's where, Sanford Health has just really had that advantage, because, the physicians are employed by us and we can coordinate schedules. I don't know how those clinics down the road do it, because they probably don't have all of those subspecialties, which again is, validation of why we're taking that measured approach. We have the right people doing the right things. And you're absolutely right. When you inject something into somebody, we have to have that gram stain and make sure that, we're perfectly safe. And then the other fun part, Peter, to me has been is that as I've learned this in terms of, real world data, and we took a lead from you, one of your articles that you wrote here, in terms of how do you really measure the impact with, some of these things. Orthopedics is, the physician generally just asks how the patient is doing, and imaging is really just not there to tell us too much about what might be actually happening with some of these therapies right now. So that's what we've designed with, this one trial that we have, as you're aware, where we're looking at osteoarthritis across several joints now, and really, asking that question in terms of the outcomes, whether it's just pain scores and mobilities. And I think that ultimately, the clinical outcomes, may be judged as not as hard a science as some of the molecular outcomes we're sometimes used to, but it's really the best way to move forward with this real world data approach. So I thank you for your efforts in that. And I thank you for always being available for these types of discussions.
Thank you. I think, we're very open here I think we've been open to trying to work with sponsors because we see that there's this potential need for individual investigators to collaborate together. Scott Gottlieb, when he was commissioner, and I put out a kind of this pathway, and then we actually articulated that in guidance too, of trying to have this collaborative development where individual practitioners could collaborate, manufacture a product at separate locations according to a similar protocol, put people into a common clinical trial protocol, get an answer, and then essentially get licensed for each of their institutions. I think we need to think creatively like this in this field of regenerative medicine, because it isn't the same as making a pill, and so we're not going to be able to export the pharmaceutical model the same way as has been done for small molecule drugs or even protein therapies.
Yeah, it's certainly, and as I think I mentioned before, with the science, the pace of the science right now, but also the pace of the consumer demand, and I think that's the bit where I really think with regenerative medicine in these clinics, I would love to see some politicians get behind this a little bit more in terms of providing you some support in terms of regulations with respect to this. The opioid crisis, got a lot of political attention, and I think the application of some of these approaches by unlicensed and maybe unqualified individuals should garner some attention as well. And getting some more physicians behind us on this would be good too.
No, yeah, I totally agree. I think the other thing that we will continue to do is try to directly educate the consumer, because I think good consumer education will help direct people to people like yourself who are doing this correctly, and help avoid kind of stem cell tourism to potentially dangerous opportunities. So thank you so much for speaking today. We could have gone on for a lot longer, but I think we're out of time now. Thank you again.
Thank you, Peter. It's been a pleasure.
Would you want to live to be 120 years old? Dr. Nir Barzilai and Dr. David Sinclair now discuss this topic with moderator Jamie Metzel.
Hello, everybody. I'm Jamie Metzel, founder and chair of One Shared World, and author of the book Hacking Darwin, Genetic Engineering and the Future of Humanity. And I could not be more excited to welcome you to this conversation about my favorite topic with two of my very favorite people in the world. Our topic today is living healthily to 120 and beyond. We all want to live long and healthy lives. We have so many examples from mythologic history of Methuselah living to 969 and so many others. But in the Bible, just a few passages after Methuselah, we have the author, who, saying, well, after now, 120 is the limit. But we have an example of a woman, Jean Calment in France, who we believe lived to be 122 with a lot of activity and a lot of health and eating 2 pounds of chocolate a week for the big part of her life. So when we think about aging, it's not about living forever. It's about unlocking human potential. I mentioned my very favorite people, and those are our guests today. Nir Barzilai, who's the Renner Chair for Aging Research at Albert Einstein College of Medicine and author of the wonderful book, Age Later. And David Sinclair, the co-director of the Harvard Glenn Center for the Biology of Aging Research and author of an also wonderful book, Lifespan. So let's just dive in. Nir, I mentioned in the beginning, this difference between lifespan and health span. I mean, it's this everyone says they want to live a long life, but nobody wants to be a vegetable. So what's the difference between lifespan and health span, and how can we measure certainly health span?
Well, the difference, all the difference is that David's book is lifespan and my book is health span, but you cannot really actually distinguish between them, because what we have found as geroscientists is that we can target aging, and if we target aging, then we delay diseases and we live healthier. And so if we delay disease and live healthier, we'll also live longer. So in fact, we're talking about the same mechanism. And if there's any difference, it's just because for many people, longevity somehow is a curse. They think that they don't think that they live healthy as they live longer. And that's the thing that we kind of have to change. We can live healthier, and the side effect is living longer. What's bad with that?
Yeah, we should be blessed with that curse. David, there's a debate in the aging research community about whether aging is a lot of different independent things, or there are some overall systemic mechanisms of aging. What is aging in your view?
Well, like all good scientists, we do debate things. That's our job. But fortunately, over the last decade, we longevity researchers, we call ourselves, have come upon a list of about 8 major causes of aging that we all agree on. And so some of these are well known. These are shortening of the ends of chromosomes, the telomeres, the loss of stem cells, cellular senescence. These cells are like zombie cells that accumulate in the body. The loss of mitochondria, which are the power packs of the cell. And the list goes on. There's one called epigenetic alterations that may be a major part of that. But science is always dynamic, and we're discovering new things all the time. But what we're really, I think, looking for here, which would be super exciting, is are there 1 or 2 major causes that lead to these other bad things that happen to our body? And in that way, we could theoretically intervene, slow down the aging process, and stay much healthier for longer, as Nir mentioned, and therefore we live longer as a result.
Yeah, that's such an important point. So Nir, you have brought together one of the largest cohorts and studied one of the largest cohorts of centenarians to learn, well, what are the lessons, the biological and life lessons from these centenarians? Can you talk a little bit about that and about the genetic lessons that we learned from that study?
Sure. So the major question is, if you get to 100, do you get your disease when you're 60 like everyone? And all you do is live longer with disease? And the answer is not. Health span and lifespan in this population goes together. They live 20, 30 years healthier than a control group. And that's not what's most exciting. What's most exciting is that they have what we call a contraction of morbidity. They spend very little time at the end of their life sick. 30% of them just don't wake up in the morning. So we can achieve that. We are built in to achieve it. There are lots of genetic component to exceptional longevity, genetic component that slows aging. And one of the major one in our centenarian is the fact that all the growth hormone mechanisms are impaired. A lot of genes of growth are impaired because we need the growth hormone when we are young to build the body. But then with aging, our body starts to break down. We have to shift the energy from growth to protection. And we can find it in our centenarians among other genes.
Yeah, it's so important. And we'll come back to that about those kinds of hacks and how different interventions can shift that balance between growth and repair. But before we talk about the kind of hacks that seem a little more aggressive, David, let's talk about the hacks that are available to everybody, the blue zone stuff, lifestyle. Can you talk a little bit about, if you want to live long, what are the really practical things that you can do? And then connected to that, in our evolutionary history, all of our ancestors have gone through these starvation funnels where most everybody else died, but our ancestors made it. Can you talk a little bit about evolutionary history and how that contributes to calorie restriction as one mechanism?
Right. Well, if there's one thing that I would recommend or suggest to live longer, it would be eat less often. We live in a world of abundance now with 3 meals and snacks. This is what it's actually doing to us, besides make us obese, is it's turning off our natural defenses against disease and aging that we have. And these are controlled by longevity genes that Nir and I and many others in the field have discovered. So you want to put your body in a state of perceived adversity. Run a little, move, lift weights, eat less often, eat the right foods, and then your body responds and fights against deterioration. Unfortunately, the world that we have is we love comfort, and this is actually accelerating our aging. So we need to really just get off our behinds, don't eat so often, and get those longevity genes to work and protect us over the long run.
That's great. And then, Nir, you've been the world's champion on studying one particular intervention, metformin, and you're targeting metformin. Your TAME initiative is not just about seeing whether metformin can work as a tool for addressing the comorbidities of aging, but establishing whether we can have the kind of systemic intervention that can pave a way for a whole field. Can you talk a little bit more about that?
So first of all, David mentions the hallmarks of aging, and he mentioned that when you target one, you actually affect some of the others. And that's what happened with metformin. Actually, we published a paper showing that it attacks all the hallmarks of aging. Why is it? It's really simple. It happens in the drugs that David developed, the resveratrol kind of drugs, and rapamycin, which is another drug. When you take the cell or organ and body and make it from old to young, you're going to change all those hallmarks. And that's what happened when you have a true, what we call geroprotection. Metformin for us is a tool to get a new indication that is a prevention of age-related diseases, something that the FDA doesn't totally grasp. Nobody really grasps this concept yet. And that's why it's so important that we're here, to show that we can prevent a bunch of age-related disease by just targeting aging more specifically. And so metformin is a drug that's safe, has been used for decades. It actually was used first to prevent flu and malaria when it was discovered that it has anti-diabetic properties. But people on metformin have less diabetes, have less heart disease, have less Alzheimer, have less cancer, and they even have their mortality of people with diabetes on metformin is less than people without diabetes. So this is an important tool to show that we can target aging, and then lots of drugs will be developed in combination, and we can really achieve what we're trying to, living healthy until 120.
Yeah, so important. And David, you are, in addition to being famous for your good looks and charm, you're also famous for, yeah, exactly, as you can see, for your information theory of aging. Can you talk a little bit about what that is and how that points us in the direction of, excuse me, of interventions that can help us live healthier longer?
So the idea really behind this is there something controlling the hallmarks of aging? Is there something that goes wrong over time that could potentially be reset? And over 25 years that I've been working in this field, working on little yeast cells that make bread and beer all the way up to humans, what the data is pointing to is that one of the major drivers of aging is the loss of what we call epigenetic information. And briefly, what that means is we have two types of information in our body. We have the genome, which is the DNA, and then we have the control systems, which are the epigenome. An analogy would be a compact disc where the music is the genome and the information in the DNA. It's very robust. And then the epigenome is the reader to read the right songs at the right time. And aging, I think of as scratches on the CD, so you cannot read the right songs at the right time. Or in the case of the cell, you cannot read the right genes at the right time. And that's what we call epigenetic decay. And that might be a root cause of aging that leads to many other problems that we've talked about. And drugs like metformin slow down the accumulation of the scratches and perhaps even erase some of them, which is exciting. But the most exciting part about this theory, if it's correct, is that the music of our cells is still there. If we polish those scratches, we can get back the youthful music of our lives. And that's what we're trying to do now at the forefront of this field.
Amazing. And so can you talk a little bit about David, about the different interventions that could polish away those scratches? You mentioned metformin. What else?
We and a few other labs work on a molecule that we naturally make a lot of when we're young. It's called NAD for short. For long, it's nicotinamide adenine dinucleotide. And it's the molecule that keeps us alive. Without NAD, we're dead in 30 seconds. It's required for mitochondria, the power packs, to work, among other many reactions. And metformin is playing a role in that. NAD is important also, not just for energy, but for regulating a set of longevity factors called the sirtuins that we've worked on for many years. And as we get older, we make less of this molecule, NAD, and we think that our defenses get less and less. And you can accelerate this decline in NAD by not exercising and eating too much. Ultimately, what we're trying to do is make drugs that will raise the levels of NAD back up to youthful levels and get the body to fight as though we'd been running marathons and eating perfectly. And those drugs are actually in multiple clinical trials across the US. There are many others, by the way. There's rapamycin near mentioned that mimics low protein diets, which seems to be very effective in animals and seemingly in people as well to rejuvenate. And there's a growing list of molecules that are either in labs or in human clinical trials that look really promising, so that metformin and rapamycin won't be the only ones on the market that we can access shortly.
So 20 years from now, when you are chronologically 20 years older and based on these kinds of interventions, biologically X years younger, and you're in your clinic and somebody comes in and you say, I've diagnosed the problem that you're facing, it's aging. And here's the treatment for aging. 20 years from now, what do you imagine that treatment looking like?
First of all, it will be telemedicine in 20 years. I'm not going to see the patient in my office. But let me take you back to a previous Vatican conference where Biden and the Pope were the keynote speaker. And Biden came, he was vice president then. And he came and he had the Moonshot Cancer Initiative. And I'll tell you, cancer is so much more complex than aging. Okay, it's really a problem because every cancer is different than the cells in your body. It's different than any other cancer. It's a huge problem. And aging is actually a quite simple thing to do. The challenges are not at all, except that if you prevent aging, you don't only prevent cancer, you prevent many other age-related diseases. So it's clearly the future. In fact, the Pope said, after Biden's talk, he said, I still hope that there's just a little pill that will cure cancer in everyone. And I think metformin is as close as it comes. It's already existing. So the only thing, unfortunately, the animal that lives almost the longest proportionally is turtle, okay? And the turtle, the problem is very slow. and really the rate is not as great as it should be, or we hope it would be. But I think that 20 years, there'll be all those drugs that have been repurposed and totally new mechanisms and new approach that it will extend lifespan very significantly. And I'm not sure that 120 needs to be really the limit, although this is kind of our maximal lifespan as a species. But, I think the future is open.
Yeah, and it's exciting because there are these types of interventions, and that's going to, it seems to me, intersect with our future of precision health and ultimately predictive health. And so the way I would imagine is that we have all of these different types of interventions, and we know a lot about the individual biology of each person, and then we're able to increasingly customize interventions. So David, we're here speaking for the Vatican. So this isn't ultimately a conversation about exciting science. Exciting science brings us to the table, but the conversation is ultimately about ethics and values. How can we make sure that our most sacred values and ethical systems, including our ancient ones, guide the applications of our most powerful technologies? And when we think about aging, there's kind of different ways that we can look at it. On one hand, if we're trying to increase average healthy lifespan globally, by far the best intervention that we can make is helping everybody around the world live lives that will allow the kind of benefits that we've had in wealthier countries over the past century, with water, sanitation, basic safety, healthcare, all of these kinds of things. So that's one absolutely necessary intervention. The other is population-wide. How can we enhance healthy lifespan? And if we do that, the inventions, the love that even a subset of us are able to deliver, I think, in my view, will help everybody. And then there are kind of, when most people think about extending lifespan, they think about crazy Silicon Valley billionaires who are pushing these frontiers. So David, is there a way to weave all of these three things together? Because we want the investment, the venture investment, the people kind of taking some big risks, making big bets, and we want to do the public health stuff. How do we, from an ethical perspective, how do we weave those three different stories together?
Well, briefly, what we need to do is enhance what we're already doing as a field. In the last 10 years, we've gone from just a bunch of scientists at conferences and really very few people listening, and thanks in part to books that are being written by you guys. The public has woken up, not everybody, but a lot of people have realized that addressing aging is the biggest impact we can have on human health by far. If you smoke, your chances of lung cancer go up 5-fold, but aging from 20 to 70 increases it over 100-fold. So aging is the major cause of not just cancer, but most suffering on the planet. So if we can address that, it would be a huge economic benefit and also social benefit. And we'd have money left over to tackle these other big problems. So this is a really global and important initiative, but we need a grassroots effort. We need more investment in research. There's very little investment into aging biology compared to cancer and heart disease. And tackling one disease at a time has actually led to problems. We can keep the heart alive for a long time, but the brain still ages. And our approach is the opposite. We want to keep the whole body alive for longer. But getting to the developing or less fortunate world, we have to make drugs available and supplements that work, that are cheap. And metformin is a perfect example of a drug that's a few cents a day, if that. And as technologies evolve, they will become cheaper, and inventions that we make in our labs will go off patent and eventually become cheap. And that's also the goal. All of my colleagues believe that we're not trying to make money here. We are trying to leave the world a better place, and that will happen.
Yeah, and that's what this is all about, is how can we, and all science is about how can we harness science, including the science of aging, to build a better, safer world for everybody, including the most vulnerable people. So we're out of time.
Can I just say, this is not all about aging only. People who survive cancer because they get radiation and chemotherapy, they age rapidly. HIV, people get their diseases 10 years
even
earlier.
COVID.
Yes.
COVID people. The people who are disabled have a problem. I would say even poor people that cannot afford to eat or exercise, they could benefit from a drug.
Yes.
And if we want to go to Mars.
And again, it's not about drug. I think that drugs are a piece of the story. It's our ability to understand the mechanisms of aging, to slow aging, to enhance health span, will not only have, as David mentioned, economic dividends, but it will have health dividends across the spectrum. So we are out of time. We could go on forever. Thank you so much to both of you.
Thanks, Jamie. It's been great.
Rabbi Dr. Eli Abadi, Cardinal Gianfranco Ravazzi, and Sheikh Dr. Asim Youssef share their insights about how we define the soul.
the scientific method is not able to identify the soul, how do we explain the existence of non-physical and immortal substance to the contemporary person?
What is the nature of the self or the nature of the soul? Is it, in fact, a non-material substance that supervenes the human body and connects with it in some way? Or is it something that is integral and intrinsically part of human biology, part of the human substance itself? And there isn't really an agreed upon answer to this question.
There used to be in the past that they used to say that when a person dies, the body is slightly less heavier than when the body was alive, and therefore that is the weight of the soul. But of course, that has been proven not to be the case.
So the question of how do we explain the existence of the soul, I would start off, and certainly from an Islamic perspective, we start off with the idea of one's self-awareness. That is to say, what you might call primary self-awareness. The one thing that you know is that you exist. Now, Descartes, in the 16th or 17th century, framed that as I think, therefore I am. Several hundred years earlier, Ibn Sina or Avicenna, the great Muslim philosopher, articulated a very similar thing in what he called the suspended man thought experiment, where he said that imagine a person has spent their entire life suspended in the air. They can't see anything, smell anything, touch anything, feel anything. They have never even felt their own body. They have no experience, effectively a blank slate person who has no experiences at all from an external perspective. Would such a person know that they exist? And Ibn Sina said yes. This person would know that they exist because of what he termed primary self-awareness. And you would start off from a religious perspective with that concept. That is a universal human concept. Now, we can understand that concept in a number of different ways. Sometimes we use the word soul. And we also go into this idea of soul as being some immaterial substance.
Il concetto di natura umana è stato nei secoli oggetto di definizioni continue e costanti. Noi sappiamo che il concetto di anima diventa dominante con la cultura greca, in maniera particolare con la figura di Platone, che distingue nettamente nell'interno della persona umana due dimensioni che sono tra di loro incompatibili, ma sono connesse. Da un lato esiste la dimensione materiale, corporea, quello che i greci chiamavano soma, il corpo, e dall'altra parte esiste la psyche, quello che ha dato origine al nostro termine psicologia, per esempio, che è l'anima, che è la spiritualità, la quale non è materiale e quindi come tale è di sua natura immortale. Non è come la materia che è legata al tempo e allo spazio, è legata all'eterno e all'infinito. E allora per questo motivo Platone, sostenendo l'esistenza dell'anima, ammetteva che il corpo è come una tomba, diceva, in greco giocava sulla parola sema, che significa tomba, soma, sema, corpo, tomba, e nell'interno di questa tomba che muore, che è caduca, c'è però questa fiamma, questa scintilla che ci connette all'eterno, all'infinito di Dio. È per questo motivo che allora si introduce questa dimensione. Dal punto di vista scientifico evidentemente non è possibile una dimostrazione di una realtà che è trascendente.
The soul is the point where the divine and the terrestrial come into contact with each other, the connection between the human and the divine. The soul, in mystical understandings of Islam, is where God's attributes and qualities manifest, such that the human soul is a canvas upon which God writes and discloses his own nature.
It is the soul that makes us understand and be able to choose between right and wrong. It is the soul that understands higher and lofty principles. It is the soul that is able to comprehend and be the recipient of the divine presence and the divine inspiration. It is the soul where the intellect really rests. We have brains, that is physical, but we cannot really have a location in the brain where intellect is. That is part of the soul.
Thank you for being with us for Day 2 of the 5th International Vatican Conference. Today we focused on the importance of dialogue and partnerships. We explored the mind, the mental health epidemic, and the impact of loneliness on health. We also spoke about the importance of preparing for a society where people live longer. We learned about the impact and the healing implications to not just the receiver, but the giver of empathy and compassion and love. And we discussed extending care into the community and the home through telemedicine and other interventions. We spoke about delivering cutting-edge therapies to people who might never otherwise receive them, and how together we can build a more equitable healthcare system. We heard about encouraging advances in regenerative medicine and treating rare diseases, and screening infants for type 1 diabetes with early intervention in order to prevent the progression of the disease. Dr. Francis Collins, director of the U.S. National Institutes of Health, discussed the links between science and faith. This was followed by a fascinating conversation from leaders of different denominations. We also celebrated 3 pontifical heroes, Dr. Andrew von Eschenbach, Manny Ahomi, and Frank Sasinowski. These truly remarkable individuals have influenced the lives of so many people through their compassion, ingenuity, and spirituality. There's another individual we'd like to honor here today, and it's a person I'm proud to call my friend, Dr. Max Gomez. Dr. Max, as he's so often called, is my co-trustee at the Cura Foundation and is an exceptional journalist who has spent his career educating the public and leading by example and providing so much hope to the millions of viewers in time of crisis.
Max Gomez's father imparted a lesson that would define Max's career. He told his son, "When people choose to work with you or listen to you, they choose to trust you. Don't betray it." Those words, originally spoken in Spanish, shaped and distinguished Dr. Gomez's career as a health and science journalist. During his years as a reporter for both local and national TV news, Dr. Max, as he prefers to be called, stayed true to his father's words. His reportorial excellence has earned the trust of millions of viewers, as well as numerous prestigious journalism awards, including an impressive 9 Emmys. Dr. Max isn't just good at communicating with experts, he's an expert himself. After attending Princeton University, he pursued his PhD at the Wake Forest University School of Medicine, eventually landing a National Institutes of Health postdoctoral fellowship at Rockefeller University. Surprisingly, his road to medical journalism had a rather non-medical start. Dr. Max was a staff DJ on the college radio station. It was only after a nudge from a classmate that Dr. Max swapped the headphones of a radio DJ for the microphone of a TV reporter.
If you've ever injured your knee, it can be hard to tell exactly what's causing the pain.
And what an impact he has made. His gift for distilling complex technical information into easy-to-follow explanations helps to distinguish Dr. Max as a consummate communicator.
Congratulations to
Well
both of you guys. Now, I don't know, how do we address you now? We have to,
Dr. Gray Hair.
Add in his signature, calm, measured delivery with its occasional touches of humor, and it's understandable why so many consider him more than just an authoritative, trusted voice.
That's the subject of ongoing, intense research.
But almost as a friend. In fact, he was recognized by Mount Sinai's Icahn School of Medicine for his flawless ability to synthesize medical research, as well as for his charm and charisma that light up the room. According to New York City's Department of Health, His coverage of the September 11th attacks helped to reduce fear and anxiety. The city honored him with its Excellence in the Time of Crisis award.
The best way to prevent getting infected.
Now, 20 years later, Dr. Max again played a vital role in reducing fear and anxiety.
Could the COVID vaccine cause infertility in women of childbearing age? Debbie, there's no evidence that the vaccines cause infertility.
This time the crisis was COVID. Carefully crafted with facts and credibility, his reports dispelled harmful misinformation, giving understanding and hope to the many living in fear of infection. His talent is also reflected on the printed page as the co-author of several books about cutting-edge medical technology. Dr. Max has a heart as big as the city he loves and calls home. Always generous with his time, he's served on multiple boards of directors and advisors, including the National Board of Directors for the American Heart Association, the Princeton Alumni Weekly, the Partnership for After-School Education, and, of course, the Cura Foundation. A tireless proponent of accurate and effective reporting, he somehow finds time to mentor journalism and medical students, as well as physicians interested in medical journalism. And Dr. Max lives by his principles. Often called upon to interview people during some of the most trying moments of their lives, when tragedy struck close to home, he was not reluctant to open up about his own experiences. He shared the harrowing story of his father's financial and medical battle with Alzheimer's, as well as reporting his own ordeal undergoing surgery for neck pain. Asked why he willingly reveals such highly personal stories, Dr. Max pointed to his father as inspiration. He taught by example, and I strive to do the same. For educating the public, leading by example, and providing hope to millions of viewers in times of crisis, we award Dr. Max Gomez the Pontifical Hero Award for Inspiration.
Thank you, Max, for your dedication, commitment, and most of all, your friendship. And congratulations to all of our heroes for their incredible contributions and leading by example. Before closing, we want to once again thank our moderators and our speakers for their participation, as well as the generous support of our collaborators, our grantors, supporters, and benefactors. They include Sanford Health, Cellularity, Akkad Holdings, the John Templeton Foundation, the Leona and Harry Helmsley Charitable Trust, Sorrento Therapeutics, Aspire Capital, Hackensack Meridian Health, the University of California, San Diego T. Denny Sanford Institute for Empathy and Compassion, United Therapeutics, Alliance Global Partners, the Guthrie Jackson Charitable Foundation, Moderna, AARP, the Eliza and Hugh Culverhouse Family Foundation, Lawrence Krimker, John and Mary Papajohn, the Produce Marketing Association, Tivity Health, the Breton Family Fund, Steve Burns, CRISPR Therapeutics, the Gary and Mary West Foundation, Healthy Villages, Horizon Blue Cross Blue Shield of New Jersey, JDRF, First Quality, Piper Sandler, Michael and Jill Solomon, Stern Ages Ventures, the Church of Jesus Christ of Latter-day Saints, Cryoport, Dr. Randall Proust, the Susan Scott Foundation, Amicus Therapeutics, Arlene and Martin Bednar, Healthscape Advisors, Malik's Pizza Palace, Selective Benefits Group, Sovereign Order of Malta, and those donors who choose to remain anonymous. We are here today because of their generosity. Please be sure to join us tomorrow at 9 a.m. for Day 3 of the 5th International Vatican Conference. We'll start with a roundtable discussion on the relationship between the mind, body, and soul as part of our Bridging Science and Faith series. This will be followed by comments from the Vatican Secretary of State, His Eminence Cardinal Parolin. We will also study the impact of the environment on our health, discuss how technology and data are enabling medicine outside of the hospital, and we'll hear from a few individuals who share new perspectives around philanthropy. We are also honored to have a special message from Pope Francis, so please join us And remember to extend the dialogue by using your social media channels to share the highlights of the conference under the hashtags Unite to Prevent and Unite to Cure. Please also remember to tag us using @CuraFDN on Twitter, Instagram, and LinkedIn, as well as @The Cura Foundation on Facebook. In the meantime, have a wonderful evening, and we'll see you tomorrow.